Hey guys, I just returned from Japan from summer vacation and we saw a lot of really cool things. We went to a science museum where we saw the famous Japanese robot Asimo perform. We also went to this park where there are a lot of deer just hanging around, chilling on the roads. So these deer are actually called Sika Deer, and my brother got lucky and he actually caught a video of two male Sika Deer fighting.
Oh my goodness! They are having a fight! It's my time!
I also learned that the Japanese word for ice cream is.
Ice cream.
We also saw a whole lot of these. So check them out. These are actually cicada nymph exoskeletons, and I find them really cool because it boggles my mind to think that one of these could turn into one of these. So that's what this video is going to be about. Now, before we talk about cicadas, let's first talk a little bit about ourselves. So us humans, we have something called an endoskeleton. We have a skeleton that's inside of us. Endo means inside. So, I mean, that's pretty obvious. We all have these creepy, spooky skeletons inside of our skin. It's encased within our bodies. Now, cicadas are arthropods, and all arthropods have something called an exoskeleton. Exo meaning outside. They don't have bones like me or you. They have an outer shell, this exoskeleton. And you can see this photo that I took of the cicada nymph is actually a picture of its exoskeleton, or what used to be its exoskeleton. Now the question arises, what exactly is this exoskeleton made of? Now, before we answer that question, I have three things that I want to show you guys first. So, say hello to Coconut Penguin, first of all. And this is the very first thing. Inside this box, I have a case with this beetle inside. And beetles are also arthropods. You can see it has this beautiful exoskeleton, just like the cicada. A friend actually gave this to me in China after I gave a biology talk. Now, up here, hanging, well, taped on my wall, is first of all my blood test. I might do a video on that later. But this is actually pens. These are pens from the insides of squids that we dissected after school one day. And these are called gladii, and they are internalized shells of squids and some cephalopods. And now for the last thing, the third thing that I want to show you guys is actually in the bathroom. So say hi to Alex, first of all. Hello. Now, this is in the bathroom because it smelled when I first got it. I sprayed on some lacquer, so it smells less bad now. This is a horseshoe crab shell. Check out that eyeball. Not eyeball. I mean eye hole, I guess. And I found this one night on Ocean City, just by the beach. That's pretty cool. Now, what do all these three things have in common? They're all made of chitin. So now we know that this chitin is everywhere. And I mean, that just goes to show the beauty of biological patterns. But what exactly is chitin? Well, on a molecular level, chitin is what's called a polymer. That basically means that it's a molecule that's made out of a lot of repeating units. And that unit, in the case of chitin, is this molecule right here. Now you might be going, wait a minute, this looks a lot like glucose, that sugar that we all know and love. Except it's got this weird thing at the bottom there, right? What's going on with that nitrogen and that thing coming off of the nitrogen? Well, because we have this NH group right here, that's an amine group, and we have this acetyl group coming out of the nitrogen, so we would actually call this part a glucose amine, and then this acetyl group is attached to the nitrogen, so this monomer, we would call this N-acetylglucosamine. It's a modified glucose molecule. Now, this is the monomer that makes up chitin. And because this is such a mouthful, I'm just going to abbreviate this to NAG, or nag. So, nag is the monomer that makes up the chitin polymer. Let's see how this actually works. So, let me just quickly show the nag as this blue ball. That's our monomer. Now, through some complex chemical reactions and enzymes and all that good stuff, we can attach two of these nags together. Now we have a dimer. And we just keep on going and going and going. And after we have a lot of these nags hooked up together, this is now a polymer. Many things, right? Polymer. And because it's the polymer out of nags, we would call this a polynag. I'm not sure if anyone actually uses that term, but hey, it's a cool term. So, what exactly makes this chitin so useful? It's because it has a lot of hydrogen bonding. So you'll remember from your chemistry class that hydrogen bonds are the strongest intermolecular force. Remember that nitrogen in the beginning? Well, that nitrogen and this acetyl group actually contributes a lot to chitin's ability to hydrogen bond with both itself and neighboring strands. So I got this image from a paper that I saw, and it's really cool because you see this extensive hydrogen bonding network that extends throughout this chitin molecule. And you can see how all these dashed lines actually represent hydrogen bonds, and it's just crazy. It's wild. It's everywhere. And this is what makes chitin so strong. This is what makes so many organisms find it useful, either for protection or as an exoskeleton or as a gladius. Chitin is everywhere. But now going back to the problem with cicadas, right? Cicadas have an exoskeleton made out of this chitin, but it's so tough, it's so rigid because of that extensive hydrogen bond network. So because it is so tough, this means that it's not flexible, and therefore the cicada can't grow once that chitin hardens. So this is why so many insects and so many arthropods have to molt. Here is a GIF of a cicada molting that I found on Wikipedia. It's pretty cool. You can see how it sheds out its exoskeleton and it literally just climbs out. It's pretty cool. Now, the actual scientific name for shedding the outer cuticle, we call it a cuticle in this case, is called ecdysis. Ecdysis. Kind of sounds like a Harry Potter spell, doesn't it? Ecdysis. Ecdysis. So, in this case, ecdysis is actually regulated by this hormone right here. It's called ecdysone. Well, duh. I mean, ecdysone is a hormone that regulates ecdysis. Now, you might be thinking to yourself, wait a minute. This molecule also looks really similar, especially this four-ring structure here, right? You've got 1, 2, 3, 4 rings. And that's because it is familiar because you've seen it before. You've seen it in things like testosterone. You've seen it in things like estrogen. Ecdysone is a steroid. That's what it is. It's a steroid. So again, we see this pattern of this molecular trend here. That's because ecdysone, testosterone, estrogen, they're all derivatives of cholesterol. Now, whenever you see an insect molt, if you were to plot, for example, an x-axis of time and a y-axis of ecdysone levels, you'll see that every time the insect molts, you'll see a spike in ecdysone, a spike in ecdysone, a spike in ecdysone. And in the very last one, sometimes you'll see a huge spike in ecdysone. So that's pretty cool. It shows that ecdysone is heavily involved in the process of molting. And then after it molts, it casts out its outer shell, its outer cuticle, and what's left is this ghostly remnant of what used to be. And we call this outer casing exuviae. A lot of fancy words in this video. Exuviae. So here we see the exuviae that I found lying on a pole. I put it on a leaf to get a better shot, a better picture of it. But yeah, this is the exuviae that the cicada left behind when it emerged as the adult form. Now, the nymph stage of a lot of insects, or specifically cicadas, is called the instar. So the instar is the nymph stage. And then the imago is the adult stage, the sexually active stage. So it's actually amazing because cicadas spend many years, I think like 17 or 18 years, as a nymph, as this instar stage underground. And only after all that time will it emerge, it'll find some kind of leaf to perch on, and then it will molt and turn into the adult, the Imago, to reproduce and see the cycle all over again.
Now, if you've been watching this video and you have some good eyes, you might have seen something in these pictures of the exuviae of cicada nymphs. I mean, when I looked around and I found like 7 or 8 of these guys, and every single one of them, I saw these white threads, and I had no idea what they were. So I did some intense research, and the answer is very exciting.
First things first, how do we breathe? Well, we breathe using our lungs, of course. But how do our lungs get air? Well, we have this muscle at the bottom here. This is a muscle, and this muscle is called the diaphragm, and it contracts and relaxes to basically change the pressure in our thoracic cavity. And that allows air to flow in and out, exchange oxygen and carbon dioxide. Now, let's go into the insect world. How do insects breathe? Or more specifically, how do cicadas breathe? Well, cicadas breathe through these things called spiracles, which are literally just holes in the side of their body. It's crazy because they have this system where air just diffuses into and out of these spiracles and through these tubes called tracheae. And then these tracheae bring the oxygen to their tissues, to their body, and then CO2 then diffuses back out. Now they have some control over the opening and closing of the spiracles, but overall this is the insect's respiratory system. Now you'll notice that these tracheae are actually made out of rings of chitin. That sounds familiar, doesn't it? Chitin is also a component of the exoskeleton, and it turns out that these tracheae are actually connected to the exoskeleton of the cicada. What does that mean? That means that when the cicada is molting, it actually brings out its tracheae along with the exoskeleton. It's crazy. That means that when the cicada is molting, they can't breathe. Studies have shown that the time before molting, cicadas and other insects, they'll actually take in a lot more oxygen. And then during molting, during the half an hour to an hour that they are climbing out of their exoskeleton, they can't breathe because their tracheae are connected to the exoskeleton and it's filled with this sort of fluid. And only after they get out of their exuviae can they start to breathe again. So it's wild. They have to hold their breath the entire time when they're molting. So that's what these things are. Those white fibers you see, that is a remnant of the tracheae inside of the cicada. And it's amazing how well it's preserved, and they actually tug it out. It's literally like ripping your respiratory system out when you're trying to molt into an imago. Crazy stuff.
Now, before I finish this video up, I just want to add some concluding thoughts. So, there have also been some studies that show that temperature fluctuations can largely affect the rate of molting of certain insects like cicadas. So, with these temperature fluctuations that are happening with things like global warming and climate change, we don't know how these might be affecting the growth rate and reproduction of these insects. And I just hope that this video gave you guys an appreciation of the beauty of cicadas and insects and ecdysis. And I hope that you guys learned something and enjoyed. Thanks.